Dr. James F. White is joining us from Rutgers, New Brunswick today, where as professor of plant biology, he and his students research the ecology of microbes that inhabit plants, that otherwise we hear a lot of as endophytes. He completed his bachelor's and master's in botany and plant pathology at Auburn University in Alabama, and his doctorate in botany and mycology at University of Texas in Austin. He is author of over 270 articles and book chapters, as well as 7 books on biology and technological use of plant microbes. Dr. White is an elected fellow of the American Association for Advancement of Science, is the associate editor or chief editor of 5 different journals, and holds a number of patents and has presented extensively in international industry and academic conferences focused on regenerative agriculture, plant biostimulants, and crop microbiome, and is highly decorated, receiving the Alexopoulos Research Prize from the Mycological Society of America, the Distinguished Research Award from Auburn University, and the Research Excellence Award. Please join me in welcoming Dr. James F. White to Rowan for today's seminar.
Thank you so much. And I just make a comment about all those things that seem like things I did. They are things I did, but that's what happens automatically when you're in this job, you're in this career. You know, you just do. You just work on research and you write papers. And it just grows, right? And then the Mycological Society, your department chair, Bentabinga, knows also that that the society supports you. You know, so you get these little things, these little awards like Alexopoulos Prize and stuff like that. So that's automatic, right? Those things come if you work. You know, if you get a job where you're able to do research, so it just automatically comes. Actually, I don't try to join any of those clubs. You know, like the National Science Foundation or this or that, because it's all just kind of puffed up stuff, really. Greg asked me to talk a little bit about my life, and I did career, and I just gave you a little piece of it, but I'll start from the beginning. My father was military, so I'm an Army brat, and we moved all over the place, ended up in Alabama, where he retired. And then when I graduated from high school there in Alabama, I went into a seminary, a Catholic seminary in Iowa. So I went to this Catholic seminary for a year and a half. It took the first year for me to figure out that wasn't for me. And then the first semester of the second year, I was explaining to the people who were there why I wasn't going to be there. And so then I transferred to Auburn in Alabama. And then I majored in, I thought I was going to major at first in agriculture because I still had the idea I was going to go save the world. You know, like everybody, a lot of young people think they're going to go save the world. So I went to agriculture to do that. And then I figured out, well, because agriculture has a lot of other stuff in it. It has a lot of chemistry there and a lot of stuff, agronomic stuff. And I thought, well, that's not really what I'm interested in. But there was this botany major that was there. And botany was more my area. So I could plants and plant physiology and stuff that I loved, right? So I went into botany and I got a bachelor's in botany there. In the course of that undergraduate degree, I got a work study job because my father was military. He didn't make a lot of money, so I was getting, work study and loans and all that, like many of you. And I got a work study job working in a lab where they were working on endophytes and grasses. And so I was, most of my time, I had a little microscope there sitting in this lab called the Peanut Lab. It was just a Quanta hut there, really primitive kind of lab. And then we were looking at these grasses. We were trying to figure out where to see these fungi. It's easy to do, and I'll have a picture of one of those kind of fungi. ones I look at, but see those. And so I got my first publication was an abstract, and it came out of that undergraduate work. And so we did that. And then I decided I like this, so I'm going to go to graduate school. And I worked on a master's degree, and I ended up actually one of the features of academic is that we tend to have highly developed egos, and sometimes that makes it hard to get along with people, especially when they have big egos too. And so I went through a couple of different major advisors, worked on my master's degree, ended up in mycology and taxonomic mycology. And so finished the master's doing that taxonomic mycology. And then I moved to the University of Texas to work with, I thought, Constantine Alexopoulos, big mycologist there. But when I got there, I found out he was sick, so I could not work with Alexopoulos, mycologist there. So I worked instead with another faculty member there, somebody named Garrett Cole, who also was a mycologist. And Stephen remembers all these people. These are all names that we know about, personalities we know about. And then in the research, I decided that this area of endophytes is really new, and there's stuff there that can be developed. So I decided I'm going to focus on that. And so I did my whole PhD. I focused on fungal endophytes. And so for 3.5 years, working on the PhD, I did this fungal endophyte work. And I completed that, got my degree, and then I moved again. I didn't know what I was going to do next, but somebody needed fossil fungus work. A paleobotanist named Thomas Taylor. And so I moved to Thomas Taylor. He had a postdoc for me, paying like $22,000 a year, which to me, that was big money at the time. Now it's nothing, but that was a lot of money. So my wife and I had gotten married pretty young, married as an undergrad. So, but together we moved there to Texas. I mean to Ohio, this is Ohio. And then I worked on fossil fungus work for a year, looked at a lot of ancient fossils, looking for fungus remains, describing what they are. And believe it or not, that ended up being something really productive. And even today, the people who are working on those fossils, ancient fungus fossils, refer to the work that we did back then, because there hadn't been very many actual mycologists looking at these things. So we were able to give some insights and discoveries and interpretations that people didn't have before. Then after that, when I was done with that, well, I figured, postdoc, that's one thing, but it's only temporary. I need a permanent job. And so I applied for a number of things. I got a job in Alabama, in Auburn, Montgomery, not Auburn, Montgomery, which is the teaching school, which was the teaching branch of Auburn University in Montgomery, Alabama. So I got that job, and I was mentioning to Greg that in the course of all the teaching I did there, I taught some 13 different courses in the area of ecology and freshwater biology and botany and a bunch of different things, right? This is what you do. That's how you do it. So, and they teach whatever they ask you to do. You go and you do that. And so I did that. And I did that. They also gave me a lab and a trailer. I had a trailer. So I could go and do stuff in the lab. And that was a great trailer. But that's a great lab. It was wonderful. But working there, it took me it was 7 days a week I was working. Sometimes the schedule went to 10 at night. It was a lot, to do that, to maintain the schedule that I had there. And then a job opened up at Rutgers University, and I applied for that, and got it. and went to Rutgers. They wanted someone to work on fungal endocytes, right? Fungal endocytes. That was their idea. So I went to work on fungal endocytes, and I have been there ever since. Working first on fungal endocytes. Then about 10 years ago, I figured out that bacterial endocytes are actually more common than fungal endocytes, and that no one really knows anything about these bacterial endocytes. So we branched out and started looking at those too. And that really is the work that I'm going to be talking about in this seminar. That, I'll refer to fungal endocytes, but because they're also involved in these same processes, and you'll see what I'm talking about when I do that. Let's see if I can make this. So I should say also, ask me questions as we go along. If anything doesn't seem clear, or if you just seem engaged and there's a hole, something that I haven't covered as well, or if you just want to check something, raise your hand. Ask me. I like to do it that way. It's better that way because then it helps me to be engaged when you're engaged too. And it helps me a lot, really. Okay, so I'm going to talk a little bit about biological agriculture, and I'll explain what that is. Basically, using microbes or manipulating microbiology to grow plants. And a little bit about that. I'll talk about endophytism, endophyte, right? Internal fights, plant. Microbes that go inside plants and don't cause disease. So I'll talk about that. And I'll talk about what will I talk about? I'll talk about this stuff. Oops. That's okay. That's okay. Oh, wait a minute. That'll work. Okay, so I'm going to contrast biological, conventional agriculture, conventional or chemical agriculture, otherwise known as conventional agriculture. What are endophytes? I've already mentioned it. I'll talk about that, touch on that. The fact that most of these endophytes are seed vectors. Many of them are seed vectors. Plants are vectoring endophytes on and in their seeds. I'll talk about that. I'll talk about this process known as rhizophagy, rhizo-root phagy eating, and it's actually a cycle, and I'll show you how that works. And hopefully, if we get that far, I'm going to talk about adaptive symbiosis. And this is the idea that plants will create symbiotic interactions with microbes that help them to survive in stressful environments. So they will, for example, if they're in a high salt environment, they'll make a symbiosis with fungi that will grow in their tissue and cause them to be resistant to the stresses to the salt stress, really. I'll talk about how fungi are modulators. Like bacteria, endophytes can be modulators of fungus disease. And I'll touch on all of those. We don't have a lot of time, so I'm not going to go into super details in everything. Oh, it's working now. Conventional agriculture, chemical agriculture, what is it? A linear process of inputs. You plant a crop and then you put inputs in, fertilizers and fungicides and whatever kind of input all along, right? And you get products. One of the side effects is environmental degradation, right? All the chemical fertilizers and chemistry we're using for our crops are going into the environment. So that's an issue. Human health degradation, environmental degradation. There's a product that comes out of that agricultural production. That product is good. Okay. If it doesn't have contamination on it, the chemical contamination is good, but it's not as rich in terms of nutrients as organic or biologically produced crops. And those crops are something like 30% a year. This biological agriculture uses microbes or uses uses methods to manipulate. Like, for example, some people put sugar on soil to stimulate the microbes to grow, and plants get those microbes and the nutrients that come from those microbes. So biological agriculture uses the microbes or manipulates the microbiome. Also, there's a lot of recycling of nutrients in this biological agriculture. Recycling of nutrients and building of soil. Soils build up when you do biological agriculture correct. And the products are richer, you have no loss or no contamination of the environment. This is I'm not a great artist, but this is this is my plant cell. This is chemical agriculture here. This is supposed to be fertilizer, NPK, right? And what do? We use this, we put this chemistry on the plants. But most of it doesn't actually go in the plant. We're hoping it goes in the plant. Some of it gets in, but only about 30 to 40% of it goes in. The rest of it goes in the environment. It does damage to the environment. Okay, so it's very inefficient. With biological agriculture, basically these are endophytes I'm talking about, mostly endophytes. And what you have here is that the plant will absorb these microbes into their cells, into the root cells, and then they will extract nutrients from those microbes. And so it's an endogenous nutrient supply. And it shows the plant cell, you see the microbes in there. And how the plants get them out of the microbes is they use reactive oxygen. I'll show you how that happens, how the plants are doing that. Show you the evidence of that. There's no law here in endogenous in biological agriculture. There's no loss into the environment. There's no damage. It's 100% efficient. We know plants like, for example, a Johnson grass, sorghum, hill, pint, that will have bacterial endophytes in them. There's a little bit of nitrogen that'll leak out of the plant, but most of it stays in. Most of it stays right in the plant. So, and the plant gets it all. So endophytes, what are these? These are microbes that go into plants and don't cause disease. This actually is a fungal endophyte. You see the hyphen there. You see this is inside of grass. This is in a tall fescue thing. That tall fescue grass. You can see the cells in the background, and you can see the hyphae right there inside. Stained blue with aniline blue. And all plants have endophytes. Generally all plants will have some kind of fungal endophyte, but all plants have lots of bacterial endophytes. And here's a bacterial endophyte in a plant root. And if you, this is an endophyte. Be careful what button. It'll forward it go back. This cell, the root cell, this is near the meristem, the root tip meristem. That's the root cell there. You see the cells there, those are plant cells, and you see in it these little clusters of blue. Those are bacteria inside that plant cell. This is a bacteria called Microbes luteus, but it makes cells in tetrads, so we can track it, we can see it, and there's nothing else, there's no organelle in a plant that looks anything like that in tetrads. Nothing that, and what we did actually to do that is this plant was a Rumex crispus. It's curly dock, it's a weed. Okay, so we took the weed seeds, we sterilized them to remove all their microbes, and then we put this Micrococcus luteus, this bacterium on it that we isolated from tomato. It's a tomato endophyte. So we put it on this weed, and the plant seed germinated, took in the microbes, and you can see them there inside the cell. They are inside the cell. And what are these microbes? They're essentially, they are soil microbes. All of these microbes come from the soil. The plant sucks them out of the soil. sucks them out. I kind of say that they absorb them. How exactly, we don't know. I'll show you images, but the process, we don't know. This hypothesis is what we have. But they're soil microbes. The plant will take the best of the microbes or take some of those microbes that work well, and they'll move them onto the seeds and into the seeds, and then there'll be seed vectors. And that's because when the seed falls somewhere, it may not be in an environment where these microbes are readily available. So it has them, it carries them with them. It carries these microbes with them. So seed vectoring is a process. Okay, this is just to show that. This is a grass seed. You see the yellow coming out of those seeds. That's bacteria coming off of those seeds. The grass seeds will have these calyx and lemons that have bacteria all over. They carry a little community on those seeds. And so that will be there for when that seed germinates, they'll colonize those roots. The plant can get them inside, absorb them from there. Also, some of these bacteria will be already around the embryo, inside the seed. And we know that because we try to sterilize plants really well, seeds really well, to remove all the microbes. And many plants can't remove all the microbes because some of them are inside. We could reduce them, but can't remove them all frequently. Now, it's possible to get most of them out if it's very old seed and they just die in the seed. But most seed is not that old, and so you'll have some microbes surviving inside. So there are two places: they're on the surface and they're inside. Okay, so the take home there is one has to be careful with sterilizing seeds, any kind of seed coatings, so it does not damage the microbes on the seeds. So, and frequently people are making seed coatings that kill the microbes on the seeds, putting fertilizers and fungicides on the seeds like that. That's a problem because the seeds are immediately compromised. They've lost the microbes that they need. So now I'm going to talk about this process whereby plants are, people have referred to this as plants are farming microbes in the soil. And they are absorbing those, they're attracting microbes, they're absorbing them into the root tips, and they're extracting nutrients out of them. They're increasing them inside the roots, and then they're pushing them out into the soils from the tips of root hairs. And I'll show you how that happens. But some of these articles that support this rhizophagy for eating process, some of the articles are here that give details, scientific details about this process. We didn't invent rhizophagy. It was something that was discovered and named by some Australians, Queensland, Australia. And this is actually the paper that they did. This paper was titled "Turning the Table, Plants Consume Microbes as a Source of Nutrients," as opposed to microbes consume plants as a source of nutrients, as in pathogenicity, right? This is nonsense. This is some figures from their paper, and what they did basically is they took a yeast and they took an E. coli bacteria, and they tagged them with green fluorescent protein, and then they put it in the plant, on seedlings of plant. Those plants absorbed those microbes, and then they could track it using confocal microscopy. This is actually from their paper. You can see a hair here, root hair here, and you see the little dots, green dots. Those are the yeasts that they have tagged. And these smaller ones, those are bacteria. And all those little things are bacteria. You can see them coming out of the hairs there. So they're all in the cells, they're in the hairs. I'll show you close-up images of that. And these investigators, this was 2010, so it's pretty recent. These investigators here, here's the lead author, Channing Pongfu, Suzanne Schmidt was the Turn Lab, and a bunch of other people on that paper. All the papers that I'm showing you are open access, and you can go get those, for free. There's no paywall in there. They're all available. Okay. So what we have added to that understanding of rhizophagy is that it's more than just bacteria and yeast that are absorbed. Bacteria and fungi, more than bacteria and fungi. Okay, it also is other organisms. Just other organisms, like in this case, it's a green alga, I think it's chlorella. I think it's a chlorella, green alga, a, a little colonial form, and you can see these around the bacteria. The interesting thing about these is the chlorella has its own endophytes. This green alga has its own endophytes. And this can also be absorbed in plants, and then the plant gets the endophytes from the green alga. You can see here's a root here, and this is a plant that absorbed these green algae, and you can see you have green algae up here. Look here, it's quenched. The green is quenching. It's becoming quenched. What's happening is reactive oxygen. The plant detects the microbe, detects the alga, puts superoxide onto the alga, and the chlorophyll is lost. And as the chlorophyll is lost, you can also see these bacteria jumping out of this, coming out of this bacterium like they're, like rats on a burning ship or out of a burning building. So the bacteria come out. The blue there is actually superoxide. We use a stain to stain superoxide. NBT, nitro blue tetrazolium for superoxide. So you can see that. That's one of the things that we do to see these bacteria, or these microbes in plants, is we use reactive oxygen stains or reactive nitrogen stains, because that's the only way you can see them. If you don't stain them, you don't know they're in there. You can't tell they're in there. So here's bacteria. Here's an example of bacteria. Here you see the root hair, and you can see the bacteria inside. That's those brown things. This is hydrogen peroxide stain that we use, something called DAP, diaminobenzoate stain. It's all the brown. You can see it because the plant is putting superoxide onto the microbe. So you make the microbes visible when you stain the reactive oxygen. This actually is the yeast. And this is the clover root hair. And you see these little dots in there? Those are the bacteria, the yeast inside that. But they have no cell walls. What happens is when the plants absorb these microbes in, they strip off the cell walls off the microbe. And so they're in there as naked protoplasms. There are actually names for them. With fungi, they have a name, a special name, and with bacteria, they have another name that other scientists have given them. But they're just protoplasms, what they are. Wall-less protoplasms, microbes without cell walls. And another thing I'll notice, you can notice here, they're coming out of the tip here. You can see right there at the tip, there's actually little pores here. You can't see them here. I'll show you the images of injections where the pores are. But you can also see they're small here. And as time goes on, they're bigger. They get bigger. They're growing. What happens is the plant puts nutrients out with those microbes, and that allows them to recover, actually. And so they reform their cell walls. They have to be bacteria. They'll also make the cell walls, and if they have flagella, they'll remake those flagella, and then they'll go out into the soil again. Okay, so the plant actually is managing these microbes. The plant is in charge of this process, controlling the microbiology at every step of the way. So this is not like a disease where the pathogen comes in, hurts the plant, takes control. The plant is in control of this process. And I'll show you how that works.
Good question.
Yeah.
Do we know how the plants strip the cell wall from bacteria?
Superoxide. Yeah, the plants use superoxide. They'll detect the microbe because the microbes are secreting ethylene, and the plant detects that ethylene. And at least this is how we hypothesize that it goes. All of this, it's all almost all at once, right? So it's hard to say for certain exactly. But what we think is the ethylene, because we know the microbes secrete ethylene, and then the plant detects that. So we've done experiments where we can suppress the ethylene, and then the plant doesn't produce superoxide. But if the ethylene is produced, you get superoxide produced. That then oxidizes the cell walls off of the bacteria, and then they become naked protoplasm. Also, and I'll point this out, nutrients are carried on those cell walls. So the plant can then get nutrients right off the cell walls. Some bacteria are also completely broken down in the plant. So these bacteria, and I'll show you images of that, even contents of the bacteria can be consumed. So there's issues here. We don't know a whole lot about it. Look at jumping. How far did it jump? Okay, now we're far. So what we have learned is that this is actually a cyclic process. Rhizophagy happens like this. Plant root tips are secreting sugars. They're putting sugars out into the soil. And those exudates, sugars, organic acids, sometimes oligopeptides, sometimes vitamins, but those actually are attracted to microbes. And so microbes come running. They're attracted to those exudates. And they come to the tip. They come to the tip there, and then the plant will absorb them into the tissues right here at the tip. And then once they're in the cells, the plant hits them with superoxide, detects them, hits them with superoxide, removes the cell wall, and begins to process them inside the cells. Degrades some of them. The ones that don't degrade, it increases their numbers. And then they will go into root hair initials. These are just little bumps on the root hair, on the root surface, right? Those little bumps. So the surviving microbes go into those bumps, and then they trigger root hair elongation. They make the root hair grow. And you see these root hairs growing, and you can see also, as those root hairs grow, the plant ejects those bacteria out of the tip of the root hairs. And so you can see, if you look at this, those are all bacteria on the tips that have been ejected out of those root hairs. I'll show you how that actually works. So the plant is, and we call this the rhizophagy cycle because it's cyclic. Yeah.
So, if a tapeworm were doing this, we would call them a parasite, right? Stealing nutrients. Is that what the plants do?
All the terminology, I would say this is a symbiosis comparable to like we're farming cows, right? That's a symbiotic interaction. Could you say that we're parasitic on the cows? Could say something like that. You know, I mean, those terms, I'm not sure those terms are useful here. I'm not sure they're useful. It is a symbiosis, and the plant is in charge. That's what's apparent with it. So it's like our agriculture. So to say that plants are farming microbes, or to say, because some people say this, plants are ranching microbes, like they're ranching sheep. You know, bring the microbes in, strip the wool off of the sheep, you know, and then put them back out of the pasture, right? Kind of similar kind of process.
Let's do that with insects. They are caring for the insects. They're caring for them, and they carry them back to their nests, and they feed them, and then they carry them out. And
No, they are. They are. They're also eating some of them. They're also eating some of them. But they're caring for them, and I'll tell you what they're doing and why your example is so useful. Because the plant increases the microbes that work with it. So a few microbes come in at the tip, right in there, many go out. Many go out. Maybe 1,000 times more go out than come in. Maybe more than that. We've never counted them. I'll show you the process. It's really cool. Plants are doing this. It's a wonderful process. Yeah.
Pretty much that word extraction. The cell wall created, and maybe there's some. We also have, for good example, I think we are a good example. The body's extracting nutrients, but those microbes colonize us and live sort of fully symbiotic within us. And they grow right as you're describing. But we're not actually shearing off the wall. And so
we don't know. We don't know that. Actually, that's correct. Paul. Absolutely. Yeah, in fact, that they're shearing off cell wall, not in the gut, right? But if they go into the tissues, and they do, and when they go into tissues, that's exactly what we see. That's where these protoplasts were first discovered. They're called L forms. They were discovered by medical scientists, the Lister Institute. The L is Lister, right? So when they find things like, for example, we're really off target, but like, for example, rheumatoid arthritis in the 60s, right? One of medical investigator looking at arthritis, looking at these arthritic joints, and he noticed these little protoplasts coming out, and then he isolated them, grew them, and put bacteria. And so the theory that rheumatoid arthritis comes from bacteria in the tissues came about. Even things like heart disease and stuff like that. We know our microbes go through our system, they go into our blood, and they circulate through the blood, and they can colonize in there. We have cells designed to get them, right? But it's not certain that they can get them all, especially if they have high sugar in the blood and stuff like that. So what is blocking us is that we haven't seen this before, right? So if we haven't seen it before, we think it doesn't work. But it may, even with animals. We know this happens with fungi, we know it happens with all plants, it's happening with algae. It may also now, animals are a different group, but we may have this similar phenomenon happening even in animals. Okay, but you're right. You're 100% correct. We don't think in the gut that we're getting cell wall stripped off. We don't think. Maybe cell death and stuff like that. We don't really know, to be honest. We don't know what happened in the gut.
I don't think used by body nutrition acquisition.
No.
So that is
Yes, this is the thing. Yeah.
Because it's symbiosis on the rich trend that I want to, which is that's a funny.
Yeah. You're welcome. This all plays good. This is a weird thing, and we never saw it before. Yeah.
What's the lifetime of these bacteria?
It's a good question. Yeah, you can determine that. And the way in the cycle in the plant, how long it takes. Okay, so you can tell if you germinate a seed, as soon as that root tip comes out, it's colonized bacteria. That's when the bacteria enter into the tip, into those cells. And then when you begin to see the first root hairs forming, that's their exit point. So we're talking about 1, 2, 3 days, maybe 1 or 2, something like that in there. So it takes that long before they cycle through the plant.
That's a lot longer than typical doubling time here. So is there something that's repressing bacterial cell division?
No,
On their
no.
You would assume that they're going to make new cell wall.
In fact, no. In fact, it's the opposite. In fact, what happens when the cell walls are removed, the plant multiplies them much more. They're multiplied rapidly. I'll show you some evidence, right? But no, they're increased faster than they would if, say, it's a 20-minute life cycle, right? 20-minute division, something like that. This is much faster than that. So, but most of this replication is probably happening inside the root hair itself. And I'll show you that. Okay. Yeah. No, I like it. It's good.
Some cycles provide defense for bacteria.
Against phagy?
Yeah, against phage bacteria.
I can't say. I don't know anything. We don't know anything about that. It probably does, because they're bombarded with superoxide, and that probably it does. And they're also dividing very rapidly. So phages might not, you know, I just don't know. You know, talking out loud, talking about stuff I don't know anything about really. So yeah, better that I not go on with that if you allow me. Okay, so this actually is an image that shows you this is a little grass root, poa, little bluegrass, weed, and this is the root tip. And you see this bacteria, this plant, this root is cultivating this bacteria right at the tip. And that's where it's absorbing them. And then it's putting them in the hairs down here. And in fact, you get no hairs if you have no bacteria. You have to have bacteria to have the hairs. So we think that hairs may not even be working the way we think they're working by absorbing nutrients. We think they're processing microbes instead, that they process these microbes. And of course, the bacteria go out here. This shows a root hair. And this is just a nucleic acid stain that we use, the cyto13 or cyto13, just bacterial nucleic acid stain. And you see the bacteria now, and they're coming out of those tips. This shows a close-up of the tip. This is a bluegrass, and you can see these little dots in there, stained a different way, but you can see the protoplast, the bacterial protoplast inside, and you can see them reforming. They're coming out right there, reforming their walls right there from the tip of the hair. You guys didn't know you would get this weird, this guy talking about this strange stuff, this weird stuff. This is the root tip. This is the root tip, and it's a branch. This is one of the plants we work on a lot. This is Phragmites australis, the common reed that's all over New Jersey. We actually have some funding to study this, controlling weeds. But you can see the tip here, you see the bacteria. You can see the right here, here. The plant is absorbing those microbes into the cells right there. Right there. There's only two places where the cell walls are soft enough to get the bacteria to go through. One of them is right here. Right at the nerve skin where the cells are being divided. The other is protected. Right there. It's soft there. And so you have entry. You have entry here. They get there. And I'll show you that process. Okay, all this does, I'm not going to go too much of this. But this is where the bacteria go in. This is the root, stained with crystal violet for bacteria. We put a bacillus in there, and this is where it went in. And you can see the bacteria are pretty much intact here. You can see the cell walls there. Crystal violet makes a dark purple right in there. They have the cell wall. You can see this is actually where the walls come off. The bacterial cell walls come off. See how angular it is? If you look at that angular, those are walls coming off of those bacteria being deposited inside those plant cells. Okay, and the bacteria, this is a close-up of it, so you can see it a little bit better. You can see the little bit of light purple or lavender there. That's the protoplasm. And you see dark, that's the cell wall, the bacteria being deposited inside. All of that, you see there, another one. There's the bacteria, and there's the wall deposited. And so it's in that zone right behind the tip that you get the extraction. These bacteria on their cell wall out in the soil, they're accumulating metals and things on their walls, actually. So when they're absorbed in, they're bringing in on their wall nutrients. Like this is actually there's a paper that biometals about calcium and magnesium binding to the surfaces. There are binding sites all over the walls, all over the cell walls, so that plants can get the nutrients out of the wall. This actually shows a root cell of what that Phragmites plant. And now you see these little clusters. These are the bacteria that are inside. These are the protoplasts. And you can see here, you can see a chain. They're dividing. They're growing. They started, you have an older one here. You can see it's not blue. It's kind of clear inside. You can see the little one here. The youngest one is blue. This one is losing protein. Superoxide goes right through the cell, clips up proteins, and the plant can get those proteins out of those microbes. But these are constantly replicating and growing inside inside those cells, the plants actually are replicating the microbes in that protoplasmic phase inside. And another thing that's happening, this is just a diagram of that. Shows the cell wall here. And there's the actual cell and protoplasm membrane out there, plasma membrane. And the bacteria go through the cell wall, they lodge into this area, and we used to think they stay out here in this little space, right, underneath the cell wall and the plasma membrane. But in fact, we now know they go actually into the cytoplasm too. And I'll show you that. But it's in this space that the plant detects them and secretes superoxide on them. So this 2O minus, that's the superoxide. It's a highly potent form of reactive oxygen, but it's short-lived. So it's just a narrow target, very potent. Doesn't last long, but it's very potent, and it'll oxidize off the film. So, the important enzyme is this one. It's called NOx. Okay, if you're familiar with animal systems, NOx enzymes are important in animals too. Eukaryotes have these. And this is an ADPH oxidase. And what this enzyme does, it will take, this is the plant enzyme, and it will take oxygen from the air, and it will, from the soil air, the soil spaces in the air spaces in the soil, take that oxygen and produce superoxide. So if you have flooded soil, so the plant can't get oxygen, the plant can't do rhizophagy cycle. It can't make superoxide. So plants don't grow well if they can't get oxygen. So they have to have that oxygen. This enzyme has iron as a cofactor, the iron. So if the plant can't get iron, it also cannot do rhizophagy cycle. So if you're using a chelating herbicide that chelates iron, for example, you compromise this natural process that plants do. Plants cannot then do rhizophagy cycle. They can't make superoxide. But this just shows. Okay, so this is the bacterium with the cell wall, maybe a rod or some other shape, whatever it is. This one has to be rod. After the cell walls are stripped off, they become these protoplasms. And as I mentioned, they're called L forms. They're known, these L forms are known to form in medical systems, animal systems, in some disease processes. They were known, plant pathologists were studying these back in the 70s. L forms were common, but they've kind of forgotten after that. And but this cell with cell wall takes a while to divide. It's like 20 minutes. These are popping out, budding very fast. You know, as soon as they replicate, they can make a new cell, basically. Take away the microbes, the plants can't grow. They can grow, they're alive, but they won't grow properly. Okay, for example, what happens is without microbes, roots don't have hair. There's no root hair elongation. If there's no microbes in those root hair initials, they will not elongate. I'll show you that. Also, the gravitropic response. Roots normally grow down like this. Take away the microbes and they don't grow down. They don't do the gravitropic response. Instead, sometimes they'll go in the air. Roots will go in the air. Sometimes they'll stay on the surface. Rarely will they actually go down. So that gravitropic response is determined by microbes in the plant. You'll have also increased root branching in plants when the microbes are there. Better root architecture, right? That's all microbial effects. Increase in root elongation, longer growth, better growth. And there's some effect on the shoots as well, because these microbes also go up. This is an experiment from an experiment. It's a root. It is a root of a Bermuda grass. We like grasses because they're tiny. We can put them under a microscope slide and look at them. What we did here is we removed the microbes. Bermuda grass species, grew it on agar, and then studied it and stained it for the brown. Actually, it's the DAB, the hydrogen peroxide stain. But you can see here's where the tip is. You can see here, there's no hairs. You can see these little bumps. Those are root hair initials. they don't elongate. So this, but in the same experiment where we put a bacterium back on, then you see this. See the root? You see the root here. They start right away. Right away. Yes, Ron?
How do you know the farming?
Well, the only way we know we didn't harm the plant is because when we put the microbes back on, they're fully recovered. So we know it's the microbe effect. The microbes are doing it. That's why. And we've done that experiment hundreds of times, more or more. You know, this actually, this is an older part of the root. You see these hairs coming out? And see the little brown dots? Those are all bacteria. All those are bacteria inside those hairs. And if you look at this, you see the little dots, those are bacteria in the cells there. There. And this is a close-up. You see the hair? And you see these little bacterial protoplasts inside. All those protoplasts. A few bacteria go in, many go out. Many go. So the plant is repopulating the soil with these microbes that work with it. Not all microbes go in. Some microbes don't go in. So it's not like they're just taking everything in. Certain respond. Certain microbes respond and they take certain ones in. Okay, this and what is it causing this root hair growth? The evidence we have is that it's ethylene. That's the one, a growth hormone. It's also a stress hormone, but it's a growth hormone in plants. It results in root hair elongation. That's well known. In nitric oxide, we detect nitric oxide in it. That also is a hormone that the microbe is producing, that causes root hair elongation. And this, and perhaps there's also other forms of nitrogen, ammonium, that are released inside those root hairs, which once they're oxidized, become nitrate. And that also causes elongation, will cause growth. Okay, so root hairs will elongate. And that, you can find details in that paper about that. Okay, this, just to show you about how these hairs are processing microbes. This is sedge. Grows in Bonaire, Dutch Antilles, this desert island. People like to go there and dive a lot. But it has all these coral formations, these fossil coral formations. This sedge grows in these little pits. No soil there, but they have sand and lots of microbes. So this is the root hair in the sedge. You can see here these little brown things. Those are the bacteria in there. Yeah. And that stain is hydrogen peroxide stain. You can see the little blue there, that's the bacterial protoplast. And you can see the brown there, the hydrogen peroxide around. And this is what it looks like above. This is from the reverse of the plate. Take the plate, turn it over, look at it, and we can see it moving, see it actually alive, and we can see what's happening. Is these hairs are processing those microbes. They're moving them around and around. This is speeded up a slight amount, but not very much. You can see this with your eye. Okay, but if you pull the root out and you bend the hair, when you pull it out, you break the cytoskeleton and you get no cyclosis. You won't get this movement. So very few people have seen this. They don't know what's happening because you pull it out and look at it, and it's like taking our body and crushing us with one of the big flat, big roller things. We can't move after that because the bones, you have to have a skeleton, right? So the plants don't either. That cytoskeleton, that's cyclosis. And what we think the cyclosis does is we think that's how the plants are breaking up the microbes and moving the microbes around, increasing them. Let me show you that. See, here's a root hair. This is the cluster of bacteria, protoplasts in there. And you can see the little ones and the big ones, little ones and big ones. They're replicating in there. And you see those. And you see some of the little ones are darker, and the big ones are not staying anymore. Because probably they're breaking down. They're actually breaking down. They're degrading. Okay, this is the root hair tip. And you can see what's happening, how this works. the plant is moving these microbes to the tip. They're accumulating in the tip. They're bumbling around here. These little ghosts bumbling around. Every now and then you'll see one get caught and get pulled back in, and goes, and you keep replicating. And these, though, at the tip, will secrete ethylene and nitrogen in mass. And that causes this root hair to grow. And when that root hair grows, it has a growth spurt. These bacteria get ejected out. And then the ones that are left on the side keep replicating like this, and then they accumulate at the tip again. They accumulate there in the tip. And then they'll, again, yeah. Yeah.
Just the images are reminding me of what it looks like when you watch polymer tube growth and sperm being like transported from the tube. So I'm just wondering if there's any, do you know anything about the mechanism of this, like that process in any way?
I really don't. We know very little about it. You know, what we know is based on using histochemical stains and observations and stuff like that, and some manipulations where we try to inhibit ethylene or inhibit nitric oxide, stuff like that, right? So, and I've never studied pollen tube growth, so I really, this is another area. If I say too much about, you'll see I know nothing about it.
It's just the movement.
Yeah, oh yeah.
And basically
Yeah.
Or the tip, like you're describing.
Well, you'll see that. You'll see that. You will see that. Look at that. Look how fat these hairs are. How fat they are. They're fat with microbes. These, you can see these that are stained. Those are protoplasts. All this cotton stuff out here, that's all microbes too. Those are the microbes. They're just not. They're not touching the membrane, or they're not recently touched the membrane, so you have the hydrogen peroxide. You don't see the staining there. Here's where they came out. Right? You can see once they come out, now they're not staining. You can see them, they're elongating. They're getting the cell wall back. That's how this works. That's how that system works. And root hairs become fat with microbes. Here's the here. You see the bacteria is all diluted. Bacteria here, with some bacteria like stuck in the small. Here you can see there's some holes. That's where some of the bacteria are coming out of the pores. That's what happens. These tips get little pores in. So when there's a growth spurt, these bacteria that are pushed out, so you see a mass of those bacteria. And this is what the model we have for how that growth is happening. And you have the root hair tip, the root hair, you have the subclosure happening, you have the bacteria accumulating at the tip, you have bacteria moving around the side. At the tip, they start secreting ethylene and nitrogen. That causes the growth spurt. Actually, well, not the growth spurt, the growth hormone causes that spurt. When that hair grows, the little pores here, these protoplastic squirt, terminology, and they're forced through those little pores, and then these come back and they're replicated in there, increased, and as this continues, suppose it happens to accumulate right through the tip, and then they do it again. In bulk, secrete growth hormone, and you have a growth of hair. You have a growth. So that's and that just keeps going until the hair, until all the microbes are gone. Okay, so this is actually what that Bermuda grass. This is a hair, Bermuda grass. We put a microbe in it from Japanese, another invasive plant, Japanese knotweed, okay, which is a dicot. This is the grass. So we moved it from the dicot to here. Didn't quite work, didn't quite fit. But you can see here, and what happens here is instead of being ejected, they stay inside the hair. And you can see them in here now. For example, here, you see that toxin, those are the bacteria that stay in, but you still had growth. It probably was a channel in the middle there, kind of like that, the little channel there. So it grew there, but left those there, and then they accumulated again there, and then there was another growth spurt, and there was a hole bacteria there. So you have a little growth spurt, and then the next tip was here, and you see another growth spurt, and like that. So these hairs are banded like that, and that's because of incomplete ejection. But it shows what's happening. Every growth spurt, bacteria are ejected, and then they build up again, and then there's another growth spurt, and they're ejected, and so forth like that, continuously until all the microbes are gone from the hair. That's tomato. That's the actual ejection process. That's what it looks like. And you can see that there are three. You look down here. I can't see it in that close. There's three little pores there that those bacteria go through. If they had their cell walls on, they probably wouldn't go through those pores. Yeah.
Is that the vacuole that's expanding? Like in number
It
2.
is, yeah, what is it? Let me tell you what it is. These bacteria are outside the protoplasm. They're outside the protoplasm. And so the vacuole is happening probably with the active physical potassium, like our cells work, right? Potassium runs in, and then you have that expansion. And because these are accumulated in the tip, they're around the edges of the protoplasm. So when that expansion happens, it then moves forward and pushes those out like that. It's, yeah, so that's how, all the signaling and all that, I can't tell you what's going on. Just kind of descriptive. These people that I'm showing, like this is the student, I'll show pictures of students who did work or gathered that in. You've already seen stuff. So we think this is a nutrient function, this rhizophagy cycle and the reason we think that is, first of all, it seems logical. Okay, that's the first, but also we can actually detect nutrients when we have microbes or nutrients plants are getting. And where are plants getting nutrients from? Right? They're getting nutrients. We've always thought they're getting nutrients dissolved in soil water. That's why our fertilizers work. The chemical fertilizers, they can get that. No doubt they can get that. So they're getting some nutrients like that. They can get nutrients from mycorrhizas. They hope we've been doing this for a long time already now, that they can get nutrients from mycorrhizas. And now we're saying they're getting oxidatively extracted from microbes, from soil microbes inside the root. And measuring that, separating that is tricky. And but we have attempted, we have tried. And one of the number of different experiments was done. I'm not going to go into everything because I don't want to go into too many details here. But one kind of experiment is the cis-enabled protein absorption experiment. And basically what we did there is we just grew some bacteria with some nitrogen that was labeled with isotope nitrogen 15, right? We can detect it because there's not that much in the air. And so we know that if the plant takes that in, we can say, well, we're taking it in. We can do analysis and measure and know that it's in. So what we did then is we put it into agarose, and then we put plants, seedlings on that with and without microbes and various other changes that we did to the system, and then analyze what nutrient, what nitrogen went into the plant. That's one kind of experiment. And okay, this just shows the experimental setup. You can see here's the petri dish. You can see the seedlings there. So they're going down. Here's the same thing. This is actually where we reduce the tall fescue. We remove many of the seedlings from many of the microbes from the seedling. And one thing to notice here, where they have microbes here, you have more of this red, brown. That's hydrogen peroxide. That's the interaction with those microbes. And here you have the reduced bacteria, and you see less of that redness there, less of that hydrogen peroxide. Because the hydrogen peroxide, the superoxide, comes in interaction with those microbes. And so you can see that. And then when we measure, if we look at, say, for example, here, this is the seedlings with their bacteria present, with their bacteria, and they get this much nitrogen 15, right? Or Or here's it with no bacteria, remove the bacteria and they get this much. It's about 30% difference. It's about a 30% difference. So the microbes were representing about 30%. Now the interesting thing, what happened when we did this experiment? We grew the seedlings without proteins in the media. And then you can see here, no proteins and no bacteria. And you get very little absorption into the plant. Here is no protein. You have bacteria present in nitrogen that was absorbed. Why is that? That's because when these bacteria were there, they could grab the nitrogen out of the air. They grabbed it out of the air. That is nitrogen use efficiency. With the microbes there, plants can then make take most advantage of whatever kind of nitrogen they can get. Okay, so that's that. It's just little, but that nitrogen came from the air. All these things were put in a canister. So I explained that they're in the same piece in a canister, an enclosed canister. So the gases, they can share the environment with the gas, right? And that's what happened here. They were able to take the nitrogen that came out of these systems into the air, whatever form it was, could have been nitric oxide or something else went into the air, ammonium, whatever it was. And then if the microbes were there, they could grab it. And that's what happened. That's how we interpret that. And one of my graduate students, she found Puerto Rico. She moved around a few times to different universities. I can't remember which one now. But what she did is she took cotton and she grew it with and without a microbe. And she got this microbe. This bacillus comes out of a wild cotton plant tree from Puerto Rico. And so she put it in and she looked at what pathways were upregulated. Okay, so and you see the pathways that were upregulated with microbes are nitrate assimilation, cellular nitrogen, heme binding, which may be important in the rhizophagy cycle, but the iron that was necessary for they're getting iron, copper, I don't know where that is. Then we have these other processes, microtubule cytoskeleton, talking about the cyclosis, microtubule-based movement. Again, the rhizophagy. So these are indications that rhizophagy have. This is just a subsampling of the pathways, right? Because when you do this, you see all kinds of stuff happening. But these are the pathways that were upregulated in the data that was published here in this Journal of Applied Microbiology. Okay, the other thing that we've done, and to test this, and I'm not going to go into details because we know it, but we used gas. What we did is we used carbon dioxide, elevated carbon dioxide, to suppress superoxide. And then we grew the plants with elevated carbon dioxide, and then also in just regular oxygen, regular air. And then we looked at the nutrients absorbed in both cases, and that we reasoned that the difference between without superoxide and with superoxide is going to be oxidatively extracted nutrients. That's what how they did. And so just going to show you a piece of that. This is for manganese. What you can see here, this is in air. This is the native bacteria, so we didn't do anything to the plant to remove microbes or put microbes on. In this case, these are all added microbes to it. But this is native bacteria. You see in the air, you get this much manganese. In the elevated carbon dioxide environment, you get this much. Huge differences. Sometimes 700% differences in the amounts of, like for example, with this bacillus number 4, look at how much you get in the air, how little you get when you suppress superoxide. And so that's all right in nutrients. What you should consider also is that plants are taking in not one microbe or two or three, they're taking in a whole communities of microbes, and they're all delivering different nutrients. So plants are able to get the nutrients they need through absorbing specific kinds of microbes. We don't know how they're selecting. We do know there's some selection going on, and some other people studying plant exits have also proposed that. plants are actually selecting microbiology based on the quality of nutrients, the kind of nutrients that they, for example, if they need nitrogen, they withhold nitrogen from their exudates. And so only microbes that can fix nitrogen can survive in that nitrogen-free exudate area. Okay, so it's having plants are selecting these microbes. Okay, I won't talk about that. And parting, because I think I have to answer it. It turns out that endophytes, that plants absorbing these endophytes are actually playing a role in adapting the plant to the environment. Okay, for example, if you look at the desert environments, the plants in the desert environments are going to have microbes that help them survive in that desert environment. And some work that was done. Okay, so the point being here is that I think we need to worry about these microbes in plants because this is how we can create resilient crops. If we get our microbes, the good microbes, back into crop plants, we could produce better, hardier crops. Okay, this is an idea actually called habitat adaptive symbiosis, proposed by these scientists, Rusty Rodriguez and his partner, Regina Redmond. And this habitat adaptive symbiosis is this, that is that the plants will create symbiotic interactions with microbes to help them survive in specific habitats. Okay, so, and that is really, that's far enough. I think you guys heard enough craziness from me today. But if I go further, it just gets crazier. So I'm not gonna, I think it's enough. Well, okay, I'll give you a taste of the crazy. This is a potato root cell. This is the nucleus. These are bacteria that are associated with the nucleus of the cell of these potato cells. And this is a hospital, and this is in the leaves of the hospital. You see the nucleus, you see these things coming out? Those are bacteria coming out of that nucleus. And they are staying for nitrogen, so the blue there is actually nitrogen. They're coming out of the nucleus and they're going to there. Here, this is staying for ethylene. You can see the ethylene right here, the purple there, when it comes out. So, like I told you, the craziness gets worse. This is a mint. And you can see the nucleus here. You see these little trails coming out. Those are bacteria coming out of it, coming out of the nucleus. We think they're in the nuclear envelope, and we think because the cells are dividing in the meristem, that's where they get colonized. And when they divide, the envelope is dispersed, right? And then after division, the envelope comes back together. And so the microbes get trapped in the nucleus. But the fact that they go in there makes us think that there's some interaction. They actually call bacteria that go in the nucleus that can control the nucleus or the DNA of the plant. They're producing things called nucleomodulants. So that's also known that bacteria that do that, produce these nucleomodulants. As it turns out, ethylene and nitrate are nucleomodulants. They cause DNA replication in the plants. And so we think that the bacteria affect the nucleus of the DNA of the plant. How much, we don't know. Epigenetics, yes, maybe more, we don't know. Okay, I think I can go into that, but I won't. So, okay, any parting, any questions? Yeah.
So you're really interested in one of the many interesting concerns. How does the plant discriminate in terms of who you're bringing in? Because you're intentionally going to
it's a good question. We have no idea. We have absolutely no idea. We know this. We know that we can take some bacteria. We take them off seeds of a plant, and we can then put them on the plant, the same plant or other related plants. And I talked about the dicot, the Japanese nut. We put that one in. It didn't quite work. We know that some of them we can take out of a plant, and they really work. They'll hurt the plant we put them in, but many of them will work fine and move them from one. But now we can also take some, put them on, and they just don't do anything. we see no growth response. They just don't go in. What is the difference? We have no clue.
What does that mean? Manufactured for mimic do you evolve strain genetic bacteria? I mean, what's it mean that you can freeloader as a bacteria in this kind of scenario, right?
I don't really know, but I'll speculate. Okay, because what happens in this process is the plant is controlling the market. Right? The plant, I'll give you an example here. They go in, the plant is hitting them with superoxide, the walls are removed, the plant is moving them around like this. The plant really is controlling everything about this microbe. Also, the plant is replicating them real fast. And that means they don't have much DNA in them, right? They may have, because they have these little circular chromosomes, bacteria, right? And so oftentimes bacteria that are fully mature will have multiple circular chromosomes. But when they're dividing this fast, when they're replicating so fast, they may not have time to develop their DNA, and they may not have time to go into any of the secondary pathways, right? They may be using only primary pathways. Now, ironically, we think of nitrogen fixation. That's actually a primary process. They need that nitrogen, so they're doing that. That's primary, right? So they can divide these cells up, and then the cells, the offspring, have to fix up. They have to recover. They got to get protein. They got to get nitrogen, and so the plant can manage that too, make them get nitrogen. We know that if we stop, for example, if we use this DAB, this stain, this hydrogen peroxide stained aminobenzidine, the one stained brown, right? We leave the plant in that, then these bacteria can recover inside the cell, and they recover, and then they'll get their cell walls. They'll get their cell walls back, so we can see them. We know they're making their cell walls back. And the other thing that they can do is they can tunnel out. They can eat a hole in the cell wall and get out. They get out. So that makes me think that the reason we don't have any disease happening is because the plant is controlling superoxide and movement, that the plant is somehow controlling those microbes. There may be pathogens. Some of these microbes in plants, things like E. coli. We could take E. coli, like the first work that they did, the Australians did. They go in plants. It's fine. Of course, that's not a plant pathogen. Okay, but other microbes might even be a plant pathogen. But if they're into the plant control, they may not, if the plant is controlling them, they may not be able to cause disease. So that's the only answer that I can give. I recognize it's an issue, though. It's definitely an issue, yeah.
So you said the microbes.
They go, the plant, mama plant, puts them they go through the vascular tissues, right? They go into the leaves, they go into ovules, we see them inside the ovules. In the seed forms, we see them there, we see them there, they go into those aerial tissues. So yeah.
So then you see them in like soap, I think.
Yeah, all of that. They're systemic in the plant. They're systemic. All plants, a lot of our crop plants still have microbes, but they lost most of the good ones. But they still have bacteria in them. You had one.
Yeah, I was gonna ask, if you decrease the diversity of microbes that are there, you can change the way that the groups form, and then if you increase the diversity of the microbe formations, like along these.
It's a good question. We've never done that experiment. I would say likely yes, but we haven't done it. Yeah.
Well, we have another class that's going to be coming in, so let's again thank Dr. White.
So long.